Hey, hi, my name's Letitia,
And I'm Sabrina.
and our project is on the discovery of A4P1W1, a fluorinated atropisomeric aryl-isoxazole acrylamide covalent inhibitor for the treatment of solid cancers. So first we'll be going through the prevalence of these aryl-isoxazole fragments as a privileged scaffold, the role of covalent warheads in drug discovery, before moving on to our 12 analog library synthetic route optimization study conducted using the LCMS, as well as some biological data before moving on to our conclusions. In the field of medicinal chemistry, privileged scaffolds, which might amplify the biological potency of a compound, are extremely coveted. And this is seen in a study conducted on penicillin G, which swapped out the amino side chain with these 2-methyl-5-aryl isoxazole fragments highlighted in red, which bear these different halogen substituents. And these three analogs, cloxacillin, dicloxacillin, and flucloxacillin, showed greater biological activity compared to penicillin G. So we were inspired by these 2-methyl-5-aryl isoxazoles, and we integrated this into our own inhibitors. And these are also seen in nucleosin, an influenza A inhibitor, as well as valdecoxib and NSAID. So moving on to covalent warheads, these have showcased tremendous success within medicinal chemistry due to their ability to form irreversible covalent interactions through the creation of these covalent bonds. And duocarmycin, which is isolated from a bacteria, is a DNA alkylating agent that binds to the minor groove on DNA and prevents DNA replication. Next is telaprevir, which was developed by Johnson & Johnson and Vertex Pharmaceuticals. And telaprevir is a protease inhibitor that binds to the NS3-4A serine protease to prevent viral replication in hepatitis C cases. Lastly is Sotorasib, and more on Sotorasib. It was developed by Amgen and FDA approved in May of 2021 for the treatment of non-small cell lung cancer. And how this works is through the GEP gap cycle. GDP is phosphorylated into GTP. However, in the case of a mutation, a GTP is unable to dephosphorylate back into GDP, resulting in KRAS being locked in this active state, leading to overactivation of downstream pathways and uncontrolled cell proliferation. And what makes KRAS G12C in particular such a hot target is that out of the KRAS-backed non-small cell lung cancer cases, a little over a third result from this mutation at the 12th residue. So these are some early efforts to for covalent inhibition of KRAS G12C. And so the Showcat lab at UCSF developed the first in-class covalent inhibitors for G12C. And this was followed up by Amgen, which produced Sotorasib, and then later Mirati. And then here are some preclinical molecules by Genentech and Novartis. And so all these molecules have in common this piperazine spacer fragment in pink and this covalent warhead in purple. And so inspired by this, we integrated this into the development of our own targeted covalent inhibitors. And we saw that Sotorasib and then the compound by Genentech both bore this methyl on that piperazine spacer. So in order to probe the effect that a methyl might have, we incorporated both a forward-facing and back-facing methyl into our inhibitors. So moving on to BTK and EGFR, here are some BTK and EGFR covalent inhibitors with the covalent warhead highlighted in blue. And of these, Ibrutinib stands out. And Ibrutinib was developed by Pharmacyclics and FDA approved in February of 2014. And it stands out among those compounds as it is potent against both EGFR and BTK. So our covalent inhibitors feature the 2-methyl-5-aryl-isoxazole fragment that we identified in penicillin, and this corresponds to the A, where A1 features no halogen substituents, A2 features 1 chlorine, A3 features 2 chlorines, and then A4 features the chlorine and the fluorine. And then we included that piperazine spacer fragment with the forward-facing methyl and the back-facing methyl. And the synthetic route for these inhibitors is a 2 or 3-step process. It's a 2-step process for the symmetric piperazines, which don't feature the warhead, or which don't feature the methyl, and those are the P1s. However, for the asymmetric piperazines, we had to go through a Bach protection deprotection step. And one thing to note about this route is it's fully telescoped, meaning we only need to perform column chromatography at the very end. And this allows us to have a better atom economy. So initially, the first step, the amide coupling, had relatively low yields, as well as using HOBT, which is explosive at high temperatures. So in order to circumvent this, we tested many conditions with different coupling agents and catalysts, and we found that 2 equivalences of HBTU and 3 equivalences of DMAP led to the greatest increase in yield, up to 93% on the A2P1 inhibitor. And so we applied these conditions to all 12 of our analogs, and we saw an overall increase in yield. And we were able to track this through use of the LCMS, liquid chromatography mass spectrometry, and where the red peak corresponds to our starting material, and the blue peak corresponds to the product.
So with our 12 analogs,
with our 12
analog library,
we ran 5 different cell lines: HT29, HT2116, 231s, Cali1, and CT26. And as you can see here, 4 of them are EGFR driven, 3 of them are BTK driven, and all of the cell lines are varies in the type of KRAS mutant. The first assay was MTT, that measures the anti-proliferative activity of our compound. Our data showed that our most potent compound was A4P1, as it was selective in EGFR cell lines, but not CT26, meaning that it may be EGFR driven. However, it is not specific against in any KRAS mutant cell lines. And we also tested Ibrutinib on the same cell lines, and it was comparable to A4P1, but at a lower potency. Next, we did the apoptosis assay because we wanted to better understand the mechanism that our compounds was acting. So we used the flow cytometer and stained our cells with propydium iodide and annexin V50. So the flow cytometer detects signals and tells us what percentage of our cells remain viable, which percent of them undergo early apoptosis, late apoptosis, and necrosis. So the data here showed that out of our compound library, most did not induce a significant amount of apoptosis, and they were under 10%, meaning that there's not much difference compared to DMSO, which is our control. In our 231s, we found that A4P1 did have pretty significant amount of effect on the cells that underwent early apoptosis and was comparable to Ibrutinib in our MTT results. We also tested whether our compounds were inhibitors of NF-kappa B, which is known to be a propagator of tumor survival as well as metastasis, and we wanted to see its effect on that, but we found that A3P2 and A4P1 did inhibit NF-kappa B signals compared to the other compounds. We also tested our compound's effect on Wnt modulation because the Wnt pathway has been shown to be also a propagator of cancer, and we found that out of all of them, none of them significantly had any effect on luciferase, except A4P1, showing that A4P1 has a dose-dependent micromolar protein inhibition. So we also tested A4P1 propanamide, which does not have the warhead, while the A4P1 acrylamide does. So we did flow on A4P1 with the warhead and also with A4P1 propanamide, and as well as the controlled DMSO, but it did not compare to A4P1 acrylamide through covalent inhibition. We also ran LDH assay to detect the amount of LDH in the cell culture media, and to directly correlate whether there is cellular damage induced by our compounds. But as you can see, the positive control has lots of LDH activity, but both A4P1 compounds as well as DMSO control were around the same level of LDH activity, telling us that our compounds may be inducing anti-proliferative activity, but maybe not apoptosis or cellular damage. We also investigated the effect of alternate alkylating warheads by further preparing for chloroacetamides that bear the different aryl fragments, which showed potent, broad, and non-selective inhibition of cell growth across the same panel of cancer cell lines. We also ran LDH assays to compare the cell death rates of each compound depending on its concentration. And as you can see here, the data shows that the higher concentration it is, the more cell death rates are seen across all three of the cell lines. However, all of the A4P1 propanamide cell death rates are lower than the others, indicating that A4P1 propanamide does not have much an effect. So comparing the three A4P1 analogs, A4P1W1 has the warhead, A4P1 propanamide does not have a warhead, and A4P1 chloroacetamide contains the chlorine. So we ran MTTs on these three A4P1 compounds to compare their differences, and we can see that the A4P1 bearing the chlorine has the highest toxicity compared to the A4P1W1, and A4P1 propanamide shows that there is no effect. So this is our heat map that summarizes all of our biodata, and so here you can see that the aryl-isoxazole chloroacetamides exhibit potent, non-selective, anti-proliferative activity, and A4P1W1 exhibits selectivity for 231 cells. We also collaborated with Dr. Bra and students, where they have covalently docked our analog library with KRAS, EGFR, and BTK. Here is an image of A4P1 covalently docked to KRAS. So in conclusion, we reported a privileged scaffold inspired design of aryl-isoxazole covalent inhibitors to key cancer targets, KRAS G12C and EGFR. So using high throughput reaction optimized by mass spectrometry, we developed a scalable prep for acylation of piperazines with aryl-isoxazole carboxylic acids, and we identified that the piperazine acrylamides had lower potency compared to the chloroacetamides, except for A4P1W1. We also observed that A4P1W1 has moderate potency in many cell lines like HT29, HCT116, and 231s, which is potentially acting through pathways that involve NF-kappa B or TNF alpha. And we demonstrated this through covalent docking.
So we'd like to thank the other members of our project as well as the project alumni, and we'd like to thank our advisor, Dr. New, and ASDRP as a whole for providing us with this opportunity. Any questions?